Thank you so much, Jim. Thank you. This is one of my favorite meetings of this year because, I was at ESMO Berlin, then came back for the Targets meeting, and I'm like, should I come back for Boston? I got like 4 invites for different Boston meetings in the next 2 weeks, and I'm like, no, this is my favorite meeting. I need to be here. And again, thank you, Jim, for fighting for the cause of rare cancers. So, and thanks, Dr. Kurzrak, for chairing this meeting, and thanks for the invite. So, all right, so why we need molecular reclassification and drug development for rare cancers? All right, live from Boston. These are my declaration of interest because of the nature of work I do. Disclaimer, the content is solely my personal responsibility, reflects my personal opinions and perspectives, which do not necessarily represent the official views of any institution or organization. Again, because of some of these slides I lifted off from my own talk in ESMO, I may have typed in tumor versus tumor. Please pardon me for that. All right, so we are seeing an evolution in oncology, right? Dr. Kurzrak showed you an amazing picture from the light microscope to molecular microscope to AI era microscope. So 20 years ago, they said we can't sequence a human genome. Then it took $3.3 billion to sequence the genome. And right now, colleagues in India, China, and Mumbai say that they can take the human genome sequencing to $1. So $1 in the next 10 years, not in 2025, but in 2035. So this is because of Moore's law, which is going down. And slowly, slow and steadily, again, many questions around AI, we are moving on quickly to the AI era. Like in no time, within the next 2 years, I think we are going to have the AI era microscope. What do we mean by the AI era microscope is multimodal data fusion, and that's coming. So we are evolving from the one-size-fits-all chemotherapy era to histology-dependent targeted therapy, to tissue-agnostic therapy. Again, you've heard a lot of talks about tissue-agnostic therapy, including ADCs, right? What are tissue-agnostic therapies? This is a different way we think about cancers. Again, to all the young postdocs out here, historically, most treatments were developed to treat a cancer that has developed in a specific organ, like lung cancer, kidney cancer, prostate cancer, breast cancer. Historically, tumor agnostic are a different class of cancer treatment that's focused on the target, not the tumor. And this perspective, commentary in Nature, published by one of my good friends, who is the current ESMO president, Dr. Fabrice Andre, talks about forget lung, breast, prostate cancer, why tumor naming needs to change. The conventional way of classifying metastatic cancers according to the organ of origin is delaying people access to drugs that could help them. The WHO put out a comment in Lancet and cited this one. So we put a commentary in Lancet Oncology, again with Fabrice Andre, the current president of ESMO, Giuseppe, the incoming president of ESMO, and my colleague and friend Benedict Westphalen and myself, about bridging tumor classification and treatment. And we're going to talk about that, why we need new molecular classification so that we can bridge diagnosis and treatment. So tumor agnostic therapies, as I said, are used to treat any kind of cancer with a specific targetable molecular alteration of biomarker. Look at these targets. These are age agnostic, right? Sam Blackman had an amazing talk on pediatric drug development. Age agnostic, tissue agnostic drug development. These are age agnostic and tissue agnostic genomic drivers. The NTRK fusions, take for instance the NTRK fusions, BRAF fusions, or BRAF mutations and RET fusions. They are age agnostic, tissue agnostic. NTRK fusions can manifest in infantile fibrosarcoma in babies or in older adults as in anaplastic thyroid cancer. Doesn't matter where the location is, it's the same fusion. Again, same thing with BRAF V600E. We'll talk more about that. Manifesting in neuroblastoma in children, high-grade gliomas, low-grade gliomas, lung cancer, melanoma. In infantile fibrosarcoma, RET fusions manifest in lipofibroma in toddlers, in children with IMT, papillary thyroid cancer in teens, lung cancer in adults, pancreatic cancer, deadly pancreatic cancers. Again, so we know about historical milestones, right? So this slide presents significant historical milestones. 1799, why this was important was Rosetta Stone. If you go to the British Museum, This stone sits there right in front of you. It was so crowded, so once after the museum, everyone left, I went and saw that for a few minutes. It was a nice moment to see that. Because why? Because we knew about the hieroglyphics, but no one was able to decipher the hieroglyphics. Rosetta Stone was key to deciphering that. That was a historic moment because it was in 3 different languages: Greek, Egyptian Demotic, and hieroglyphics. With the Greek, they could decipher the hieroglyphics. 1861, Archaeopteryx. This was a transitional animal. 1859, as you all know, Darwin wrote On the Origin of Species. But there was no animal or organism that could prove the link between a transition, like dinosaurs and birds. So Archaeopteryx fossil was discovered in 1861, and I saw that in the Berlin Museum a couple of weeks ago. And this was a major event, historical milestone, that buttressed Darwin's theory of evolution. Cinema by Lumiere brothers, right? They used to watch the PEEP kinetoscopes, what Alva Edison invented, but the Lumiere brothers premiered the cinema that we have now. 2017, again, the first ever tumor-agnostic approval of pembrolizumab, because for the first time in oncology, a drug was approved based on a genomic biomarker, not based on a tumor type. That was a historic milestone. So it's been almost a decade, and we have more and more approvals. 2022, September 21st, TargetCancer Foundation meeting and gala happened. I think Jim knows that. And why this was important? Because that was also the same day where we got tumor-agnostic approval of my favorite kid on the block, selpercatinib, for RET-positive cancers. Again, tissue-agnostic. 2023, November 15th, TargetCancer Foundation meeting. Same day, repotrectinib, rare cancer, rare biomarker approval. 2024, November 20th, TargetCancer Foundation meeting. Zanidatamab, I think my jazz colleague Elena is here. I didn't know you were coming. But it was approved for HER2-positive biliary tract cancer. Again, same date. Again, I think Jim picks the date when a drug is going to be approved or vice versa. All the stars align. So 2025 was also historic because for the first time ever in any major meeting at ESMO, we had a specific track called histology tumor-agnostic track. Had the pleasure and privilege of chairing this track. Again, I screenshotted this if you want to believe that. So again, think TargetCancer meeting, 21st, Boston, again, selpercatinib approval. November 20th, again, zanidatamab approval, 2024. November 15th, 2023. Again, we got this picture. We'll keep our fingers crossed. We need to force EMA or FDA to approve something. Something needs to happen, all right? So why was also 2025 important? Because of this ESMO, 50 years of ESMO, right? For the first time ever, we had a tumor-agnostic track in any major meeting, right? I've been trying to talk to many of the organizations to have a specific tumor-agnostic track because they have colon cancer track, lung cancer track, right colon cancer, left colon cancer, but never a tumor-agnostic track. But first time ever, we had a tumor-agnostic track, and we had a concurrent publication in Cancer Discovery, right? AACR Cancer Discovery published this, my personal perspective, which does not reflect the any organization or any institution, published in Cancer Discovery. Please take this QR code. I would like you to read this publication and reply back to me. Please agree, disagree. But these are my personal perspectives on the interface of drug development and tissue-agnostic drug development. I'm sure you would have all read this book by Malcolm Gladwell, Tipping Point. It was written 25 years ago. He talks about how little things can make a big difference. So the FDA approval of pembrolizumab was a tipping point for the field of tumor-agnostic precision medicine. Why? Because a drug was approved for a genomic biomarker. It was also important because it represented the marriage of two fields, genomics and immunotherapy. Approval was not based on randomized controlled trial. Approval was based on retrospective real-world data. Again, age-agnostic, tissue-agnostic genomic approval. Since then, we had not one, not two, but 10 different tumor-agnostic approvals. Again, this is all listed in this recent cancer discovery paper, and this is also listed in what the FDA and EMA have approved many of this. So since then, we've had pembrolizumab, larotrectinib. We have 3 drugs approved for NTRK fusions. Larotrectinib, entrectinib, and repotrectinib. Latest kid on the block, June 2024. Pembrolizumab, once again approved for TMB high cancers. Dostarlimab, again, Dr. Andrea Sesek showed amazing responses with dostarlimab. That drug got approved for DMMR positive cancers. Dabrafenib and tremetinib, we'll talk more about that, got approved for all BRAF V600E positive cancers. Again, the first ever targeted combination therapy. And then selpercatinib on May 2024, initially for lung cancer, and then in 2024, 2023, for 2022, at the same time of Target Cancer Foundation meeting for all RET fusion positive cancers. Again, we are in the era of ADCs. Oncology is a fashion industry, right? Again, it was TKI, immunotherapy, now it's ADCs. And ADC, we have the first ever approval, and I want every ADC to have a histology agnostic indication based on a biomarker, and more soon. So NTRK, not one, not two, but 3 different NTRK drugs. We need to find these needles in haystacks. Let's talk about the second book, The Black Swan. This is a book by Taleb, and he writes about, the book focuses on extreme impact of rare, unpredictable outlier events and the human tendency to find simplistic explanation for these events retrospectively. Taleb calls it the black swan theory, and I call it my BRAF story. June 2022, dabrafenib and tremetinib got approved in a histology agnostic manner for BRAF V600 positive tumors in a tissue agnostic space. As you can see here, BRAF tumors are age and tissue agnostic. Again, can be seen in many, many cancers beyond melanoma, thyroid cancer, and colorectal cancer, and in pediatric cancers as well. Hairy cell leukemia, it's 100%. Question is, can we all put them in one basket? Yes, I say we can all put them in one basket. So in one of the first ever histology independent studies to be designed early in 2010 and 11, this was called the WEM basket study. Simple idea, any patient with any cancer, metastatic relapse refractory cancer comes into the clinic, we do testing for BRAF V600E. If they are positive, they get on this study. They were on different baskets, lung cancer, cholangiocarcinoma, all others, including rare cancers, and another rare cancer called Erdheim-Chester disease, Langerhans cell histiocytosis, deadly cancer, anaplastic thyroid cancer, and colon cancer as well. So again, we saw tissue agnostic sensitivity across all solid tumors. And there was a rare cancer called Erdheim-Chester disease, Langerhans cell histiocytosis. Based on this cohort, basket study cohort of less than 25 patients, we were able to secure an FDA approval because biomarker-driven rare unmet need did not require a randomized controlled trial. So we were armed that this could be done. This was the first study that also showed that although colorectal cancer did not respond to monotherapy vemurafenib, the combination of cetuximab and vemurafenib led to the response. So this shows the pan-cancer activity that was published in Cancer Discovery of vemurafenib in BRAF positive non-melanoma cancers. Again, clearly you can see these waterfall plots for the non-oncology audience here. Waterfall plots show the shrinkage of the tumors. Below these dotted lines, 30%, that means it's a response. So if there are 2 scans showing a response, it's confirmed response. Clearly you can see the color coding. BRAF is a tissue agnostic target. Many, many tumors with different tumors shrink to this. But although we were buoyed by the success of this targeted therapy, unfortunately the agony of failure, every 4 to 6 months, every patient universally progressed. Science moved on. We had combination therapy in melanoma called BRAF and MEK that became standard of care. So we designed the next generation of the basket study. Again, simple idea, we call the ROAR basket study. Why ROAR? Because I'm a major fan of Lion King. True story. My next protocol is called Mufasa that combines BRAF, MEK, and immunotherapy. The idea is simple, rare oncology agnostic research. Patient with a deadly cancer like anaplastic thyroid cancer, biliary tract cancer, glioblastoma comes in. As long as they harbor the BRAF V600E alteration, they get 2 drugs, dabrafenib and tremetinib. every one of these cohorts, look at this anaplastic thyroid cancer cohort. So the non-oncologist here, right? Anaplastic thyroid cancer is a very, very deadly cancer, one of the most lethal cancers known to mankind. If there is a patient who lives beyond 6 months with this diagnosis, we call the pathologist to ask them if does it have any papillary or any other component in it. That's how bad the disease, deadly the disease was. We started talking to our endocrine colleagues. They started testing patients. And the more patients we tested, the more patients we found with BRAF heredity. The first ever patients to get on this study was the family member of one of my colleagues. So because we tested him, we found that patient got in the study and had a complete response. So this is a waterfall plot of that anaplastic thyroid cancer. 69% response. And again, at that time, it was owned by GSK. I think they had an ad board. And this was like around at least 10 years ago. I was much junior. No one listened to me. I said, this is great. This is great. We don't need anything else. We don't need a randomized study. We just need to go and get an approval. Again, all the experts in the room said, no, we need a randomized study, 50 patients with this drug, 50 patients with standard of care. I said, there is no standard of care that exists. And I convinced, and then this was acquired by another company. So I convinced the new company to directly go to FDA and ask them. FDA said, hey, this is good data. Come back in, median survival time is 6 months for this tumor. Come back in 1.5 years, which is 3 times, with more patients. We'll consider that. No randomized study, control trial was done. The drug got FDA approved. Data set, less than 25 patients, non-randomized study. So again, this shows the power of biomarker-driven drug development in rare cancers. I showed you several arms. Each and every one of those arms read subsequently. Cholangiocarcinoma, 50% responses. I asked the company to see if we can go get an approval in cholangiocarcinoma. They said it's going to be hard because all these are rare cancers. For every approval, they need a team. They need a whole team. It costs millions of dollars. And the 100 or 200 patients that are there existent, may not be feasible for them to develop this team. So with all the data sets, we did a pooled analysis of this road basket study, all the adult studies that were published, and at the same time, the NCI match study published their study as well. So we got data from that, and we did a pooled analysis of all the pediatric, adult, and road trial, and we applied for FDA approval. After 20 meetings, we were able to secure a tissue-agnostic approval for BRAF. Again, BRAF, as you know, is always considered an exception to tissue-agnostic drug development. Again, the colorectal cancer put a wrench in the development. This is why we need a molecular reclassification of cancer. Let me tell you why. BRAF is a tissue-agnostic target with tissue-agnostic approval because in the real world, patients will not have access to treatments without an FDA approval. How many patients can go to niche boutique academic centers or clinical trial units to access these drugs? So this was my black swan moment, the impact of the highly improbable. But think about that. The date of approval for this drug in melanoma was May 29, 2013. But the date of approval in a tissue-agnostic space was June 22, 2022. I remember this date very well. We were in Banff. I think my better half is here. She knows. And I didn't have cell phone access. So I had to be on national television media at Banff. So we exactly remember this date. So, but it took 10 years, almost a decade, for a patient with cholangiocarcinoma or brain tumor with BRAF V600E alteration to have access to the drug in the real world. This leads to molecular inequity. The BRAF mutation in melanoma, that patient has access to treatment, but the BRAF mutation in a glioblastoma, that patient did not have access for 10 years. This is just one biomarker. Think about all the hundreds of biomarkers that we have and all the hundreds and thousands of patients that miss out on these treatments just because of an anatomical accident. So personally, I believe that drugs should be approved based on their molecular alteration, not based on an anatomical alteration. So we learned our lesson for RET. So we were able to secure an FDA approval May 28th, 2020 in non-small cell and thyroid cancer. September 21st, TargetCancer Foundation meeting and gala happened. At the same day, we got an approval of tissue-agnostic approval of red positive. Like 2 years, we were able to apply the lessons learned in the BRAF into this study. I'm showing you these waterfall plots. Pancreatic cancer, cholangiocarcinoma, every cancer. But there is a story behind each one of these waterfall plots. There is a patient and a family member. Take, for instance, that pancreatic cancer patient that had a 100% response, complete response. Meet Alison Cuban. Again, she's my patient. And she progressed on standard lines of therapy. I still hate to call it standard lines of therapy because we don't cure cancer based on the so-called standard lines. And when she presented to my clinical trial clinic, she did not have hair. She had fluid was full of, belly was full of fluid. And we did the sequencing. We found red fusions. I was so excited internally. I thought she hit the genetic lottery. We have a drug to go after that. But it still wasn't FDA approved. This was still on a clinical trial. Had to maintain my compassion, composure. And I said, let's start the trial tomorrow. She said, no, I'm going to get married in 2 weeks. So I'll come back after that, and we'll get started. I knew she may not make it beyond 2 weeks. So I said, why don't you get started on the trial and go get married? She started that study. 3 weeks later, she sent me videos from her wedding, her dancing in her wedding. Threw her wig out because she was young. She started growing hair in the 3 weeks. Ascites vanished, out of wheelchair. These are the stories that keep a clinical trialist, a drug developer, a molecular oncologist, an oncologist up and running to clinic every morning. We enter an era of ADCs. Again, I don't want to spend a lot of time. We had a great presentation about Enhertu, trastuzumab deruxtecan, showing amazing responses, and it's now approved in a tissue-agnostic manner. But there are many unknowns in drug development, right? There are challenges in tissue-agnostic drug development. One size does not fit all. There are many unknowns. So again, how can we tackle these unknowns? So ESMO, we put together a group. Again, I co-chaired this group with the ESMO Precision Oncology working group called ESMO Tissue-Agnostic, Tumor-Agnostic Classifier and Screener. Again, amazing group. So what we came up with definitions. We came up with when a target is tumor-agnostic, when a target is tumor-modulated, when a target is tumor-restricted. Tumor-agnostic is a target like NTRK or RET, where it is agnostic across the board. Tumor-modulated is a target like BRCA, when it can be modulated in specific tumor types. Tumor-restricted, I would say, like EGFR mutation, that's specifically seen in lung cancer, that's not seen in many other cancers. So also we propose a tumor-agnostic drug development framework for investigators in industry. How do we tackle biomarker-driven drug development? And we show how we can have a biology-informed mechanistic rationale, early regulatory advice here, adequate trial design, followed by confirmatory evidence. So we need to enter a paradigm of not one size fits all, right target, right biology, right patient, right drug, clear benefit. We need a drug approval. We need to move away from the one size fits all. Again, I need to thank ASCO for inviting us to think about designing studies in rare cancers. And we put this thing together. I think it's one size does not fit all. And again, clearly you can see in pediatric AML, all these diseases are treated as one. But think about that. These are different diseases. And you can see the analogy with the sweater on and the colors on, right? So we need to move away from the one-size-fits-all approach in oncology. Again, we spoke about AI. AI-driven drug development is going to unravel many more tissue-agnostic targets. The timeline of drug development today is 10 years or 15 years from we discover the drugs. I think personally I would say this is completely unethical. We need to be fast, we need to be quick. So, Malcolm Gladwell wrote Revenge of the Tipping Point. Again, this is a fascinating book as well. 25 years after that, he revisits the tipping point. And what is the revenge of the tipping point for us in precision oncology? So there was a rare paper published when they sequenced 300 tumors, 300,000 tumors. They found 21.5% with tissue-agnostic indication. Only 1/3 of patients with NTRK fusions got eligible therapy. We have 3 drugs approved for NTRK fusions. And the challenge was, are we picking these needles in haystacks? We are picking these needles in haystacks, but we are not threading the needle. Why is that? Because of access barriers, knowledge gaps. Again, I still give a lot of talks in the community. Many oncologists are not aware. We don't have a genomically savvy workforce. Many times patients bring it to them. And most of them are tested in a therapeutically futile stage. You see the prevalence of tissue-agnostic targets. 21.5% of all metastatic cancers had a tissue-agnostic target indication, including 5.4% of patients that lack a cancer-specific indication. So that is why we need a new molecular classification for cancer. We need to bridge diagnosis and treatment. What are the next steps? We need to reframe classification around molecular drivers. We need to adopt unified precision medicine framework. We need to build a next-generation oncogenomic savvy workforce, redesign clinical trials and regulatory adoption, bridge diagnostic therapeutic silos, promote policy and cultural change. And thanks to TCF for organizing this meeting. This is like cultural change. Today, I have a call to action. We need a molecular reclassification of cancer. We need transformative acceleration of tissue-agnostic drug development from today's 10 approvals to 50 to 100 within the next 25 years. If I had my way, if I had power for an executive order, this would be it today. Just crushed it at TCF, the greatest cancer meeting in history. I hereby sign the most revolutionary executive order ever. Total molecular reclassification of all cancers. This is bigger than the moon landing. We are not just bridging diagnosis and treatment. We are obliterating an old system and building something spectacular. Tumor-agnostic treatments are going to be so powerful, so incredible, we are making them great again and greater than ever before. This is Nobel Prize territory. I'm asking for that. Maybe 2 Nobel Prizes, peace and medicine. Complete total victory over cancer is coming. The big, beautiful breakthrough in medical history is coming. Many people are saying that this is the greatest achievement, possibly in all of human history. Cancer is finished. We are winning. How do we get there? Again, we need to think about strategy and logistics to get there. 5 strategic imperatives mentioned in the cancer discovery paper. Genomic testing as standard of care, not last resort. Building molecular fluency in clinical practice. Revolutionizing clinical trial design for molecular medicine. Transforming regulatory frameworks for molecular precision. Real-world evidence as continuous learning engine. I have a vision for precision oncology and reclassifying tumors. When? As JFK said, we chose to go to the moon, if not as who, if not now when. The call to action is 50 to 100 drugs within the next 25 years based on molecular biology and molecular alterations. Please read this. I also give a checklist for tumor-agnostic drug development. Please take this QR code. Again, I want your honest opinion. Please agree, disagree with this publication. We academics, we need debates. And Europe, I think Europe group put together an amazing white paper as well. Again, this is a QR code for that white paper because many drugs are approved in the US and not in Europe. So this white paper was put together with the ESMO meeting. Then of course, thank you. Thank you everyone. Thank you, Jim. Thank you, Dr. Kurzrak, and thank you everyone for being here. And hopefully we can climb Mount Everest one day. Perfect time. Q&A. All right, we'll take Q&A. Yes.
So beautiful talk. So I'm going to challenge you. The issue that I think a lot of clinical oncologists have is that we have therapies, standard therapies, that work. Wait. But we also have then NGS that will show, say, an NTRK fusion protein. What is your opinion on sequencing?
I think that's a great question. My personal opinion versus what is happening in the real world. Let me talk about my personal opinion. I wouldn't call the therapy standard. Again, that's my personal thing until we have a 100% cure or response for that particular drug. So if there is, say, for instance, an NTRK fusion, personally, I would go frontline with the NTRK fusion because we need to hit it. Again, these therapies, I mentioned age agnostic, tissue agnostic, and I would say it should be line agnostic as well. Line agnostic approval in the sense the oncologist should know when to give these treatments, which is in the first minute, first round. And again, there was a recent paper, pediatric oncology paper, that showed that NTRK fusions, 96% responses in pediatric tumors with NTRK in the frontline setting. And again, the relapse refractory setting was like 70 to 80%, frontline setting 96% responses. So all these therapies could work much, much, much better in the earlier lines of treatment. Dr. Andrea Sersek showed this morning using dostarlimab in dMMR cancers, 100% responses in rectal cancer. I think if we get all these precision medicines to earlier and earlier lines, I think we have the potential to transform medicine in an unprecedented manner, obliterating what we are seeing right now.
What's very interesting, because there was an experience in breast. Jennifer Litton did a study in in primary breast cancers carrying a BRCA mutation. She did a pre-operative study of a PARP inhibitor called talazoparib, and it induced pathologic complete responses. So it worked, no chemo in BRCA positive. That went nowhere.
Because when you cure a cancer, you stop giving the drug and it's not commercially viable. True, but we need to do, again, this is a question again asked. I think commercial regulatory reasons should not be why we do drug development. I can say in a patient advocacy community that we need to do drug development which is patient-centric. We need to move away from drug-centric to patient-centric drug development, and that will fit together. Again, the reason why we are here is for patients. I think patient-centric drug development should be why we do this. Question? Yes. Last question. Thank you.
Yeah, fantastic talk. Thank you very much.
Thank you.
How are we going to get there? So if we need 100 approvals, we need 100 targets. So what do we have to sequence, tumor, liquid biopsy? What do we need to do to identify these targets? Because if you are sequencing in our panels, we're never going to see new targets, right? So how do you envision this technical part to identify the targets and then to get the drugs?
Again, as I said, we need to have a comprehensive molecular portrait of every patient walking in the door. And if you can put the slides, I can show the biomarkers. Again, we don't need to look for new biomarkers. For the current biomarkers, look at that. For ALK, we have 6 different drugs approved in lung cancer, or 7 different drugs approved in lung cancer, ALK fusions. And ALK fusion lung cancer, you have 7 different drugs, and 3 more drugs going to be approved. So in the next 2-3 years, you will have 10 drugs for ALK fusion positive lung cancer. But if that same ALK fusion is seen in an ovarian cancer patient, we can't prescribe any of those 10 drugs. So just because the ALK fusion is in lung cancer, you have 10 drugs. Just because the ALK fusion is in a sarcoma or ALK fusion is in ovarian cancer, you don't have a drug to prescribe the patient in the real world, right? So that is molecular inequity. I think as a group, again, I have the strategy, but we need to talk about logistics on how we get there. So these are the biomarkers that we have. For ALK, BRCA, EGFR, FGFR2, 3, we need tumor-agnostic approvals. Again, what's happened is all these tumor-agnostic drugs you can see are designed as an afterthought. But what we need to do to get to the 100 over the next 25 years is we need to do concurrent tumor-agnostic drug development together with tumor-specific drug development. Right from the get-go, right from the first minute, first round. We shouldn't be waiting for a drug to be approved in a primary indication, waiting for it to be, other tumors to be added on. That takes a decade. And in that time, hundreds and thousands of lives are lost during that time.
Great. Thank you very much.
Thank you. All right, one more, and then we'll stop. Yeah.
Hi, thank you. It's a great talk. Shelby Doyle with Alliance for Rare Cancers. I'm wondering if you could speak a little bit to what you mentioned about the need for the real-world data and continuous learning systems. It seems in the U.S. system, because we don't have centralized health systems, that's a real challenge. And AI is obviously an opportunity, but it also makes data an asset, right? So with your colleagues in the EU, they've been a little bit more ahead in thinking about sharing that type of data. What thoughts do you have on where we should start in the U.S.?
I think that's a great question. And one of the things that we were discussing with regulatory and academic and industry colleagues is, for many targets, like for instance ALK, there is data for approval, but they have data in like 10 different tumors. What about the 10 different tumors that were not enrolled in the clinical trial? So the key was the regulatory authorities were okay with approving the drug based on the 5 to 7 different tumor types, and then using real-world data as supplementary evidence later when it comes in. So Dr. Kurzrak wants to answer. Yeah, he has a comment as well.
We reviewed ALK, and we called it the next tissue-agnostic approval. I think the paper is probably about 2 years old now, but the issue with ALK, actually, for approval is that there's a lot of different ALK inhibitors, and the different diseases are with different ALK inhibitors. Mm-hmm. And so that's another barrier because, so do you approve all the ALK inhibitors? Like personally, I would say yes, but I don't think the regulatory agencies would agree with that. Or do you pick one of them and approve it across the board? But when you look at the different, it's clear that multiple different diseases, and by the way, this could be hematologic disorders as well as solid tumors. We could cross that bridge. But on the other hand, it's like 6 different ALK inhibitors. So which one gets the approval? And I don't know if you have a solution for that.
Again, that when thinking about the different algorithm, not the drug getting approved. So one thing about ALK is there is one drug that has tumor agnostic data, nice 60% response data, but the company is not interested because it's not a viable market, maybe 100 to 200 patients a year. It's not a viable market. So some of the investigators from Australia and Japan reached out if the investigators can file for approval in their own countries and also to FDA. And there is no mechanism in the US for investigators to approve and approach FDA. It has to be through a pharma company. And Japan has that mechanism. I think it would be approved, hopefully. It could be approved in Japan because there is a very motivated investigator who's pitching it to the government. It will be a political approval, not a regulatory or clinical approval. We will see. And Japan will lead the way, and hopefully we can follow that. So to add to that, age agnostic, tissue agnostic, line agnostic, zip code agnostic as well, and country agnostic as well. Thank you.